Good morning, everyone. Nice to see you all so animated. Here we are in our final week of lectures for the ecology module. The final lecture I have set up, I think I called it conclusions or something like that. What I intend is to finish up anything that I missed, but that shouldn't take but a short amount of time. And then what I plan to do is review, I plan to review points of confusion or things that I can clarify, things that I know students would like to have reviewed because you've asked me questions on email or in office hours. Things like the Ali effect. Things I'll build a few slides for and just do a brief review of. And then we can also have some question and answer on material from throughout the module. Please come to that session. It's going to be your review for the exam. And you're bound to learn a lot from that session. I'm not sure it's going to be webcast. All the more reason to make sure you're here, right? So that's concerning a week from today, Monday. In these final 3 lectures, particularly today, I'm covering material that's not necessarily in the text at all. So this lecture and portions of the remaining lectures, the most important material for you will be the PDF of slides and the webcast. Much of this material just didn't make it into the book. All right. I'm going to continue right along with, are we rolling upstairs? Are you all by yourself today, Dave?
Yeah.
Oh, sorry. Take your time. All right. I finished last time by talking, no, actually I didn't get to it at all. But I gave you the slides for these cycles, these water and nutrient cycles. And I'm going to ask you primarily to rely on the material in the book. I'm not going to go over them in detail, but I'll cover them briefly here. That's a continuation of last lecture, but today's lecture is about paleoecology, a word I don't think that even makes it into your textbook. I checked a couple of other ecology textbooks, and the word didn't even make it into those. And yet this is a major subdiscipline within ecology, and I'm going to try to give you a sense of what it is today. It's something I'm very much interested in. It's something that many people in this department work on, paleoecology. And what we're going to try to do today is think about some of the principles we've learned in ecology in a deep time context, in the context of geological time, ancient history, and draw forward to today, gradually this week. That's what we're going to do. We're going to work from the deep past through to the very recent past to present-day dynamics, using principles we've learned throughout the module. One thing I mentioned briefly were the use of isotopes in ecology in a previous lecture. I'm going to do a little more on that today, and particularly because of the importance of isotopes and trace elements and biogeochemical techniques in paleoecology. Then we're really going to start to take an anthropocentric focus. We're going to focus more and more on the evolution of our kind, the group to which we belong, because our next lectures are on human ecology. So we'll start by looking at some ecological mysteries of the early Pleistocene in Africa and move forward toward the end Pleistocene when we had large-scale extinction dynamics around the globe, and we'll look at the possible human role in those dynamics. So from last time, we were, where I was trying to leave off was with a review of global cycles of water and elements. Matter is cycled throughout ecosystems, across ecosystems, within ecosystems, on a global scale. And you all know the story of how you follow the little raindrop as it falls from the cloud and hits the mountain and makes its way into the stream and then into the river and back to the sea, only to be evaporated and go back into the sky and do the round again. That's the hydrological cycle. It's more complex than that. It's driven primarily by solar energy and evaporation from the oceans, evapotranspiration from the land, transpiration from the plants, evaporation from the land. And it's of major importance in global ecology, given the importance of water to living organisms, of course. So your book does pretty well in briefly summarizing these cycles. And I'd like you to just understand their dynamics, the basic reservoirs that contain the major elements of interest. For nitrogen, well, let's start with carbon. This one will be particularly important in our discussions of carbon dioxide in the atmosphere. That's the primary atmospheric reservoir in gaseous carbon dioxide. It's taken up by plants in the process of photosynthesis, and please consult your book for how it proceeds from there. Nitrogen, you're not responsible for the details of the nitrogen cycle. It's just would require too much memorization for current purposes. So just the very most general contours of the nitrogen cycle, and then understand the phosphorus cycle. It's quite straightforward. One of the interesting differences is that phosphorus does not occur in any significant gaseous form in the atmosphere. So it's a primarily terrestrial and aquatic-based cycle. These are elements, right? These are atomic elements: carbon, nitrogen, phosphorus. Those are elements of major importance to living organisms. They're critical to the growth of the vast majority of living organisms. There are other elements that are either less critical or critical in lesser amounts, or that get taken up by organisms but really aren't essential for their biology at all. Elements that exist in nature, perhaps in trace amounts, that may not be important for the functioning of organisms, the biological functioning and physiology of organisms, but they get taken up by organisms in the things they eat or the things they drink, and they get lodged in their tissues. As a result of that, we can use these elements as natural tracers of ecological dynamics. And the relevance of it here is that we can study these elements as they move through these systems today, but we often have access to these elements in fossils that record the ecological dynamics of ancient organisms. So by understanding the dynamics of these elements today, and by studying fossils, and fossil soils, but bones, yes, teeth, yes, plant tissues, yes, but the soils themselves and the rocks that we find fossils in, we can understand something about ancient ecosystems. Just to give you one example of how trace elements and their measurement can shed light on the structure of communities. And this is from a classic study in the High Sierra, 3,000 meters. It's a lot higher than what's indicated here. This is way up in Yosemite. And in this study of Elias et al. In the early 80s, they went to that canyon, a pretty sparsely vegetated canyon. Certainly you have good soils in some of these pockets, and you have various plants growing and mammals and birds and insects living, but it's a pretty simple community compared to, say, a tropical rainforest or something like that. And what they did was they sampled a bunch of plants, focused on the sedges from this genus. When we see SPP like that, we just mean species, multiple species, or more than one species. So when we see that notation there, we mean genus Carex, a couple species or a few species of Carex. They also looked at tissues of voles, microtus, should be capitalized. You can see that I made these slides on the fly, huh? And then the major consumer of the voles and one of the top predators in the system, martens, Martes americana. So here you have a simple trophic chain, and of course the plants themselves are getting their nutriment from the soils and picking up some elements from the waters that they imbibe. What the investigators focused on were strontium and barium, non-essential elements for biological growth, but the elements that resemble calcium in a physical sense and thus get taken up by organisms seeking calcium. Because calcium is what organisms are after and require so much of, they tend to favor the uptake of calcium relative to strontium and barium. This is what's called the biopurification of calcium. So an organism tends to have less strontium and barium. Organisms have less strontium and barium along a trophic chain as a result of the biopurification of calcium. You don't really have to understand the details of that. What you really need to recognize is that as you look along a food chain, from the sedges to the voles to the martens, relative to the soils and the rocks from which the plants grow, you see a decrease in this ratio of strontium to calcium and barium to calcium as a result of the biopurification of calcium, as a result of the preferential uptake of calcium by organisms. There you have a quantification of a food chain dynamic in this simple system based on a SRCA and BACA ratio. What if you could get these ratios out of fossils? You might be able to start to reconstruct fossil ecosystems and the ecology of fossil organisms. And indeed, with some effort, you can get these data from fossils. Isotopes we mentioned in the context of ecophysiology and redwood's use of fog. And I said you weren't required to understand the chemistry behind it. Let me just point out that these are stable isotopes, though. You'll be introduced probably to carbon dating in the evolution section, radiocarbon dating. You've all heard of that. That's based on radioactive isotopes of carbon that decay over time. The ecological use of isotopes is based on stable isotopes, isotopes that do not change, do not decay over time to any appreciable degree. An isotope such as these isotopes of nitrogen also tell you a lot about trophic level in some aquatic systems. So here we have a shrimp and a small fish and a big fish. And we would have this shrimp consuming zooplankton, and those zooplankton consuming phytoplankton. And what we see is, as we move up consumer levels, we see a very consistent shift in nitrogen values. Very similar to what we saw with SRCA and BACA ratios. Just another example of how we can measure an element on a mass spectrometer and have it shed light on ecological structure in the living world. And with some applications, this also works in the fossil context. So let's look at some fossils and think about what we can know about them. I'm going to favor some of my favorites, mostly talking about mammals here, partly because we're leading into humans, but also just because that's what I know more about and what I like. But we could do something similar with other organisms. So here we have a fossil. You can probably see something happening here, right? Can anyone make out anything of interest here? Can anyone see any resolution in this fossil slab from China? Somebody said vertebrae. Some vertebrae there. Can you see anything else? Paleontologist sees a lot when they look at that. That's so interesting. It's not just a broken, what's that? A hand-like structure? Yes. See the head here? Little teeth. Foot here. It's part of the long tail. But look at the impression around the tail. That you might not be able to see from where you're sitting, but these fine striations and this impression around the tail is actually quite remarkable. It's preserving the hair of this ancient animal. And this is well over 100 million years old. This is from dinosaur times, right? This guy was living in the Mesozoic, the Middle Jurassic. And with painstaking work, all these little pieces can be found and described and mapped out. This is an organism that's never been seen before. Nothing's ever been seen like this. So a paleontologist looks at it and pieces it together based on other known organisms, but tries to keep an open mind because this is something that's never been looked at before. What do we do with it? I mean, we have all these pieces. We can use just the anatomical form of this organism to start to reconstruct it. In biology, as in other parts of the world, form fits function. The form of something tells you something about its function. It's true in architecture and as it is in biology. So if we know something about the form of the femur on this creature and the structure of its hind foot and the structure of its tail, we might be able to say something about how those things were used, about how this animal functioned in its environment. And that's good old-fashioned comparative anatomy with an ecological emphasis. It's an ecomorphology, if you want. It's using the morphology, the form of this creature, to reconstruct its ecology. And so from that slab, based on a lot of work, these authors reconstructed a creature something like this. Yeah, aw, that's cute. And they named it Castorocauda lutris similis, otter-like beaver tail thing. Yeah, because it apparently had this big flat tail. This has nothing to do with beavers. This has nothing to do with otters. This is way before anything like a beaver or an otter evolved. But it had a tail kind of like a beaver and probably used it for propelling itself through the water in conjunction with those big splayed out hind feet that had a partial webbing between them, as well as long claws. And then these teeth that were kind of otter-like had a curvature that suggested they might be good for grabbing slippery prey and holding onto them. Thus, otter-like beaver tail thing, right? And, maybe they're way off base. Maybe this thing lived in trees. Probably not. I mean, this is a hypothesis. This reconstruction is a hypothesis, but it's sufficiently narrowed that you can expect relative degrees of truth here. But the more we study it, and if we were able to get some chemical signals out of the teeth of this organism, say, based on what it ate during its life, if those signals are still preserved, you might be able to make a much more firm reconstruction of this creature. Here's another one. This one was found in the western US by a former Berkeley student. And that's what they had to work with, this three-dimensional mess. You have to jigsaw puzzles if you're going to be in this business. So you get that mess and you start to tease it apart and then piece it back together and use comparative anatomy and other lines of evidence, and you get a little creature like this. They name it Frutafosser. Kind of built like a tiny armadillo or a tiny aardvark or something, based on its teeth, those little tubular teeth that look a lot like an armadillo's teeth or an aardvark's teeth, and those big, heavy, scratching foreclaws suggested that it was probably a digger. And based on the teeth, a consumer of social insects, ants or termites, a myrmecophage. Pretty good reconstruction. You'd like to see other lines of evidence to test it. and to prove it wrong or to support it, or to weaken it, I should say. We don't really, we very rarely can prove something wrong in biology. We just weaken something to the point where it becomes irrelevant. But absolute disproof rarely exists, just as absolute proof rarely exists in biology. These are hypotheses that face ongoing test. Another favorite of mine, because it kind of turned the tables in our thinking on Mesozoic evolution. This is Rhipenomammus, beautifully complete specimen of this organism, a mammal, found. Big for this time period. Most of the organisms living in the Mesozoic around the time of the dinosaurs were fairly small. Not as small as they used to be when they were thought to all be mouse-sized or smaller. But this guy was, the size of a house cat or a heavy house cat or something like that, an opossum or something like that. Big teeth in those jaws, big heavy skull, clearly a predator. And what was it eating? Well, the preservation is so good that the stomach contents are visible in this organism. That's a special view on the ecology of this organism. Well, let's look in its stomach and see what it ate. Forget the chemicals and trying to interpret those. Let's just look in its belly, and you can see that in the belly it had these. It had another organism in there with a whole different skeleton. And what was in there? Well, there was a little dinosaur in its belly. So this ancient mammal had been eating a baby Psittacosaurus, eating a little dinosaur, reconstructed in this way with Archaeopteryx or something like that up here on this branch. And that kind of turned the tables for some people. We didn't tend to think, we thought of the mammals as being the ones underfoot of the dinosaurs. But here you have a relatively large mammal consuming a dinosaur. And that has a lot of relevance to the way we think about the food webs in the Mesozoic and the structure of those communities. I think I made that word up, so don't expect to be understood if you use the word dinosaurivore. People who study dinosaurs will say, big deal. What's the big deal here? It's just a little, it's whatever, But if you're into mammals, that's pretty cool. Mammals eating dinosaurs. What are these? Primates, great apes. Yes, they're both. And they're our closest living relatives. Orangutans, gorillas, chimpanzees, bonobos. Wish I could talk about them, but really what we want to do here is talk about, we want to move forward and talk about early hominins. You'll get some of this in the evolution section. But a hominid is a reference to the big group, including the great apes, as well as ourselves and our closest ancestors. I'll give you a term for just a reference to ourselves and our closest relatives to the exclusion of the great apes in a minute. That's hominin, and I'll show you how to spell it on the board here. But let's just look at some of these forms, and this is from a Scientific American article from 20 years ago, so take these reconstructions with a grain of salt. But here we have some of the Australopithecines, shown as walking on two legs, good bipedal things, long arms, protruding jaws, hairy is how they're portrayed here. Obviously different in certain basic respects from the great apes. Great apes don't walk on their hind limbs, generally. They can do it for brief periods of time and in particular environmental contexts. It's always of interest when they do it, but they don't do it habitually. And these forms did, based on an analysis of the hips and the knees, all the way up to the necks and the musculature of the whole system. These guys all walked on two legs, walked on their hind legs. In some of them, the digits were relatively curved and long. They probably did well in the trees also. So they were doing well on the ground and in the trees. They're small creatures. So this one, Robustus, one of my favorites, about 4.5 feet tall. or 40 kilos, maybe 100 pounds. That's pretty tiny, right? It's like Nicole Richie or something. It's pretty small. The later one, they get bigger over time. So Homo, our genus, represented in here, is, ourselves, Homo erectus here was very tall, very heavy boned. Neanderthals were massive, perhaps related to the cold climates they live in. You often see animals just becoming bigger in colder climates, or stockier in particular. And they had huge heads, bigger brains in their absolute size than many of us. We vary tremendously in the size of our brains, and it's no clear reflection on intelligence. It's related to your body size pretty strictly also. And Neanderthals had massive brains to go along with their relatively massive bodies. So there's something of a tendency to get taller and bigger and heavier in time here. Not a major trend, but there's definitely a strong trend toward increasing brain size, and very interesting trends relative to the teeth that reflect the diets of these organisms. With form-fitting function, the teeth in particular, as the site that food crosses on the way to the gut every day, those teeth reflect very clearly, in so many cases, the diet of the organism, the physical properties of the diet of the organism. So let's just take a quick view of the hominin fossil record. And there's your term for the organisms more closely related to ourselves than to chimpanzees and bonobos. So this is just our closest group, hominins. It's a group within the hominids, a bigger group. And here we are back to 7 million years ago now. This is an extraordinarily fast advancing field of study. An unbelievable amount has happened in the last 15 years. It's been a heck of a couple decades for paleoanthropology. And no institution on Earth has contributed more to this record than Berkeley. Berkeley has a very long-standing history of study of hominin evolution. And in the last 30 years has made the biggest contributions to this field, bigger contributions than any other single place, by far, really. Ardipithecus is one of the most recent. Tim White and his group and their discovery of Ardipithecus in Ethiopia, not the oldest hominin, but one of the most complete, all the way back to 5 million years ago. This Australopithecus group has been known for much longer, and there are a few species of Australopithecus. It just means southern ape, something like that, because it was first found in South Africa. Great stories behind all of this, and I urge you to try to learn more about it. Very interesting history of discovery, and just, interesting creatures. These are creatures so close to ourselves. This is a group that I'm very much interested in, the robust Australopithecines. So they evolved from Australopithecus stock, but they derived particular features of their skulls that make them look quite different. They're massive, the skulls. They're heavy, they're robust, and the dentition is different. And for me, this is a really interesting ecological question about, well, what were they doing ecologically? Made all the more interesting because they coexisted in time and space with Homo, with early members of our genus. So they lived in the same places. You find the fossils together at sites in Africa in this time period, from around 2.2 and change to 1 and change. For over 1 million years ago, for over 1 million years in the Pleistocene, you find robust Australopithecines occurring with Homo as fossils in deposits in Africa. The interpretation is that they lived on the same landscapes together. What's a kind of a red flag that throws up if you have that kind of evidence in an ecological context. Yes? Competition, absolutely. The competitive exclusion principle, all the way back to Gauss. And this has been a major topic of discussion because these things have been known about for 60 years. The coexistence with Homo has been argued for that long and has been assumed for 40 years. So everyone thinks they live together. Well, then how did they persist for so long? They must have been doing something different ecologically, suggested all the more by the really unique skulls of this group. And a very interesting skull here as well. A much more gracile construction in this group, a much more lightly built construction. The teeth are much smaller in our line than in this line. The brains are bigger in our line. At the time of origin of the robusts, that's when you get the first clear evidence at about 2.5 million years ago for the consumption of large mammals, for butchery, for the butchering and consumption of large mammal carcasses, something like a big antelope like this. And this is Berkeley, these are Berkeley workers who discovered this. They discovered bones at this age with marks on them, these striated marks that when magnified, and you can look at the internal structure of these marks, they're clearly cut marks made by tools that you sometimes find in the same deposits with these bones. The earliest stone tools date from almost exactly this time as well. And here's strong evidence what these tools were for. This is the shaft of one of the legs of an animal like this. And this is its jaw with cut marks inside the jaw. And you would break up a bone like this perhaps to get to the marrow inside, the fatty, nutritive marrow inside a bone like this, or to scrape the meat off of this bone. Or they interpreted these cuts inside the jaw as perhaps the removal of the tongue of the creature. It's good to eat if you can get it in terms of its nutritive contribution. It's not clear who did this. All you find are these bones and the tools and then a scattering of fossils in the area around the same time. But you don't find the tool in a hominin's hand and then the belly of one of the hominins with the diet in the belly like we did for Rapena-mammus. So you've got to piece all this together with the bits of evidence that you're given. Here's just a distribution of hominin fossil sites in Africa with a focus on those that have this coexistence between the robust types and the early Homo types. You can see that they coexisted across this entire area in Africa. We don't have many fossil sites at all from this area, in this rainforest zone, or the desert, the great deserts. We don't have fossils from there. So in some ways, we're just, our focus is narrowed as a result of where things preserved in the Rift Valley and in cave sites in South Africa. So that's a major limitation. But clearly from Ethiopia to South Africa, these things coexisted. I'm not really going to give you the details on this, more just hope to trigger some of your thinking and maybe have you investigate it further. But let me just show a couple pictures of these robusts. These are big teeth. These are much, much bigger teeth than you have in your mouth. That's very thick enamel. That's the enamel band right there. Your enamel would just be a little sliver of this, the enamel that coats the dentin or dentine of your teeth. And that enamel this is one of the hardest biological substances known in nature. It's an extremely resistant substance. Why this major enamel load on these massive teeth where your canines, this is a canine incorporated seemingly into this back grinding row of teeth. Your premolars and your molars and your canine all seeming to share this grinding function. They were clearly masticating something that caused a lot of wear on their teeth, and they served to resist it by making their teeth heavily enameled and creating this huge surface area for that processing. This is a specimen from one of the most recently discovered specimens from South Africa. There's one of the skulls. I just don't have time to go into the details of the features. But one of the other neat things about these teeth is they're so smooth. You often find them, they're puffy like this. Someone I heard said they look like little marshmallows. It's like you took one of your teeth and inflated it with air. And you have these puffy cusps, and they wear relatively flat, but with these smooth bulbous surfaces. They're very pretty things, and it's just a fascinating question what these guys were doing. And it's exercised anthropologists for quite a long time, and there are about 6 or 7 major hypotheses for how they lived. This was a traditional one that maybe they were pulling grass and eating grass seed, like these types of baboons that still live in Ethiopia today in the highlands, gelada baboons. Well, no, maybe they were cracking nuts with the back teeth like this, like these South American monkeys, and you just had to have these big teeth to get access to the food inside. Well, for various reasons that didn't fit, particularly in relation to the chemistry, the biogeochemistry and the isotopes. So maybe they were kind of like these swamp-dwelling gorillas that were eating aquatic vegetation and wading out in the water and eating the roots of these sedges and grasses that live near wetlands. Or that doesn't quite fit all the evidence. Maybe they ate bamboo, something very much like bamboo. Bamboo does grow in Africa, not to the extent that it does in Asia, where pandas eat it, but it can be found. Maybe they were just some kind of hardcore vegetarian like this, and that's always been the basic assumption, that they were a vegetarian creature. If you notice, all these models, which are formal models in the literature, are all vegetarian models. But it didn't fit all the evidence, so maybe they were just great omnivores. Maybe they ate everything, and like pigs or bears or something, just ate whatever they could and ate a lot of it. Well, that doesn't really fit the tooth structure. I mean, in a very basic sense, hominin teeth and human teeth look like pig teeth and bear teeth, but the details are not the same at all. Omnivores don't have that smooth, puffy construction. So the party continues, and we continue to search for good tests of ever more clear hypotheses for what these organisms did. At some point, those robusts went extinct around 1 million years ago, a little earlier than that. No one really knows why. Some people point the finger at Homo sapiens, the big-brained, tool-using, coexisting competitor, potentially, of the robusts. It's easy to blame these guys, earlier forms than this, for the extinction of the robust Australopithecines. For whatever reason, the Homo line continued, and the robust line died out. And there was extremely rapid cultural and anatomic evolution in the Homo line, leading to our species, Homo sapiens, at around 160,000 years ago, if you want a single date to hang on it, based on discoveries, by Berkeley teams in Ethiopia. A reconstruction of the fossils found from this 160,000-year-old fossil ancestor are here. Here's a skull just from Europe, classic Cro-Magnon man, right? With this globe of a skull and the big brain you can imagine inside. This is a big factor in human evolution, is this ever-enlarging brain, not only because of its relation to intelligence, but because that thing's really expensive to drive a brain like that. To feed a brain like that is a challenge. To grow a brain like that and keep it fed and nourished is a big challenge and requires a very dynamic ecology to sustain it. So all these things are of great interest. I hope you'll follow up on some of them. At some point, humans got out of Africa. Hominins got out of Africa. It happened much earlier than we originally thought, probably. As early as 2 million years ago, hominins were starting to move out of Africa. But hominins didn't get to the Americas until practically yesterday. It was so recent in geological time. around maybe 12 or 13,000 years ago in California. And maybe not much earlier than that, anywhere in the Americas, anywhere in the New World. The classic idea is they walked across the Beringian land bridge connecting Asia to Alaska at times of lowered sea levels. Maybe they took boats along the coast and went island to island, island hopping. Maybe they came over the North Atlantic. Lots of debate on that subject, of course. However, it occurred. People arrived in the Americas very recently and then exploded in the Americas, found it altogether to their liking, moving quickly from Alaska to Florida to Tierra del Fuego. And in California, here at the end of the last ice age, you can see a reconstruction at about 18,000 years ago, say, of the big glaciers that would have occupied the Sierra Nevada, this incredibly rich Central Valley network of wetlands draining the mountains out through the bay, and an extraordinary shelf of resources off the coast with the kelp forests and the abalone and the mollusks and everything else. So quite a place to live. It has been for the last 18,000 years. Here's a diagram of species richness of mammals in the state. The more mammal richness here than any other state in the country. The vegetation's diverse, the mammals are diverse, the geology's diverse, and people diversified here themselves. 64 distinct languages were documented by anthropologists, and that was, that's a lot was lost to history there. So it's hard to know exactly how many distinct cultures and peoples there were in the state, but incredible diversification within a short amount of time. Some of the things these people encountered we don't see anymore today. For example, mammoths, including dwarfed mammoths, pygmy mammoths, living out on the Channel Islands. There's a little pygmy mammoth next to a person compared to some other extinct elephants or living elephants. Tiny things, almost you look them in the eye. And people would have encountered these things when they arrived in California. Organisms like that on those islands, or the big ones on the mainland. Here's a comparison of the pygmy to the big one, something like Mammuthus columbi from the mainland. It's just to show you some of these elephant teeth. I'll just carry on in the interest of time. Really big bison that would have been in the Central Valley there. Smilodon, of course, saber-toothed cats. This is your state fossil, if you weren't aware of that, that your state even had a state fossil. There's a saber-toothed cat sculpture over by the geology building. This cat is found in many deposits in California. Ground sloths, these are big. These are bigger than bears. Giant sloths that lived on the ground and walked on all fours around the ground, right? Not just hanging in trees up there nibbling on leaves. Giant ground sloths, and lots of them all over the country and all over South America that people would have encountered of various sizes. Some the size of like a small bear, others massive creatures. All of these things, all of these creatures, all of these very large mammals went extinct at the end of the Pleistocene. Of course, we have some large mammal survivors here. We'll take a look at a couple of them. But these extraordinarily large ones, your proboscideans, your elephants, your giant ground sloths, camels. This place was full of camels, camel relatives. A llama is a type of camel. Don't just think of a humpback dromedary camel or something like that. These things tended to go extinct in a short amount of time, and the timing of that extinction is strikingly correlated with the timing of human arrival. Not just here. Humans arrived around the world at different times in different places. So our arrival into, say, Australia occurred at a different time than it did into the Americas, somewhat earlier, maybe 60, 65,000 years ago. It occurred much later on a big island like Madagascar, maybe within the last few thousand years in Madagascar. But in each of these places, you have this uncanny correlation with the loss of large vertebrates. And that correlation suggests causation to many scientists, that humans maybe had a hand somehow in those extinctions. And this is a great debate that goes back to the 1800s and is very much alive right now. Here's just collections of herbivores and carnivores from the La Brea tar pits, where many of you have probably visited in Southern California. Look at what was living in California. Big camels like this, bison, horses, and then maybe more familiar pronghorns and deer. Giant bears, lion-like cats, cheetah-like cats. We had a cheetah. We had an American cheetah living in this country. Wolves. And so what's left in terms of the big mammals? Well, not even the grizzlies are left, technically, in California, because they were all extirpated in the early 1900s. I think the last one was shot in 1922 or something like that. Maybe they'll be brought back at some point, no doubt politically complicated. But when this Berkeley biologist studied bears in 1910, he could recognize, based on museum specimens, he could recognize 7 different types of bear. Subtle differences, same species, but he called them subspecies, within California. It's thought that there was about a 13-to-1 person-to-bear ratio when Europeans arrived, in California. 130,000 native Californians, 10,000 grizzlies in California, 13-to-1. And of course, we remember this history of bears in the state in various ways, and they're all over our seals and flags of the state. Bear Republic, right? But how about wolves? As late as 1850, wolves from San Diego to Sacramento. And again, around in the early 20s, the last wolf extirpated from California. This is a species like the grizzly bear that still survives today, just not in California. These losses are easier to document than those previous losses. People shot them out. People hunted them. People altered the habitats, and people shot them. And that's why we don't have them now. Less clear are those end Pleistocene extinctions. 2 more minutes, you guys. I always lose my train of thought at the end when you guys start wrestling. I'm like a flight attendant up here saying, stay in your seats. You're more than welcome to leave like 5 minutes early, and I'll just say this for the benefit of the professors who follow me. If you leave 5 minutes early, it's really not a distraction at all. You might have to get all the way across campus, but leaving, when you start to stir, it doesn't sound like much when one of you is doing it, but when 35 of you are doing it, it's quite a rumble. And I simply lose my train of thought. So that's not going to help you understand this stuff. So if you need to leave, just leave a few minutes early and go for it. Otherwise, please stay put. I don't keep you late. So just stay put until the end. Thank you. So this is really the last thing you need to understand about these extinctions. It's a graphical depiction of what I've been trying to say, which is that of the large mammals in these different places, North America, Australia, South America, the ones that went extinct in the dark shade tend to be the large types. Mammals that survived are shaded in the light color. Those that went extinct in the dark colors, this is size, biomass on the x-axis here. So the ones that went extinct are all concentrated in the largest size categories. At a place like La Brea, you find a bunch of lizards and mice and even insects. I think there's one toad that went extinct. And then all those big mammals we were looking at went extinct. One toad out of all the birds and maybe another bird or two. But basically, none of the small things are going extinct. So why indeed that type of discrepancy? If it were climate change that were causing this, you might expect more general extinction. We can talk about it a little more next time. Thanks, folks. Good morning, everyone. Nice to see you all so animated. Here we are in our final week of lectures for the ecology module. The final lecture I have set up, I think I called it conclusions or something like that. What I intend is to finish up anything that I missed, but that shouldn't take but a short amount of time. And then what I plan to do is review, I plan to review points of confusion or things that I can clarify, things that I know students would like to have reviewed because you've asked me questions on email or in office hours. Things like the alley effect. Things I'll build a few slides for and just do a brief review of. And then we can also have some question and answer on material from throughout the module. Please come to that session. It's going to be your review for the exam, and you're bound to learn a lot from that session. I'm not sure it's going to be webcast. All the more reason to make sure you're here, right? So that's concerning a week from today, Monday. In these final 3 lectures, particularly today, I'm covering material that's not necessarily in the text at all. So this is water and elements. Matter is cycled throughout ecosystems, across ecosystems, within ecosystems, on a global scale. And you all know the story of how you follow the little raindrop as it falls from the cloud and hits the mountain and makes its way into the stream and then into the river and back to the sea, only to be evaporated and go back into the sky and do the round again. That's the hydrological cycle. It's more complex than that. It's driven primarily by solar energy and evaporation from the oceans, evapotranspiration from the land, transpiration from the plants, evaporation from the land. And it's of major importance in global ecology, given the importance of water to living organisms, of course. So your book does pretty well in briefly summarizing these cycles. And I'd like you to just understand their dynamics, the basic reservoirs that contain the major elements of interest. For nitrogen, well, let's start with carbon. This one will be particularly important in our discussions of carbon dioxide in the atmosphere. That's the primary atmospheric reservoir in gaseous carbon dioxide. It's taken up by plants in the process of photosynthesis, and please consult your book for how it proceeds from there. Nitrogen, you're not responsible for the details of the nitrogen cycle. It's just would require this lecture and portions of the remaining lectures. The most important material for you will be the PDF of slides and the webcast. Much of this material just didn't make it into the book. All right. I'm going to continue right along with, are we rolling upstairs? Are you all by yourself today, Dave? Yeah. Oh, sorry. Take your time. All right. I finished last time by talking, no, actually I didn't get to it at all. But I gave you the slides for these cycles, these water and nutrient cycles. And I'm going to ask you primarily to rely on the material in the book. I'm not going to go over them in detail, but I'll cover them briefly here. That's a continuation of last lecture, but today's lecture is about paleoecology, a word I don't think that even makes it into your textbook. I checked a couple of other ecology textbooks, and the word didn't even make it into those. And yet this is a major subdiscipline within ecology, and I'm going to try to give you a sense of what it is today. It's something I'm very much interested in. It's something that many people in this department work on, paleoecology. And what we're going to try to do today is think about some of the principles we've learned in ecology in a deep time context, in the context of geological time, ancient history, and draw forward to today, gradually this week. That's what we're going to do. We're going to work from the deep past through to the very recent past to present-day dynamics, using principles we've learned throughout the module. One thing I mentioned briefly were the use of isotopes in ecology in a previous lecture. I'm going to do a little more on that today, and particularly because of the importance of isotopes and trace elements and biogeochemical techniques in paleoecology. Then we're really going to start to take an anthropocentric focus. We're going to focus more and more on the evolution of our kind, the group to which we belong, because our next lectures are on human ecology. So we'll start by looking at some ecological mysteries of the early Pleistocene in Africa, and move forward toward the end Pleistocene when we had large-scale extinction dynamics around the globe, and we'll look at the possible human role in those dynamics. So from last time, we were, where I was trying to leave off was with a review of global cycles of water. Too much memorization for current purposes. So just the very most general contours of the nitrogen cycle, and then understand the phosphorus cycle. It's quite straightforward. One of the interesting differences is that phosphorus does not occur in any significant gaseous form in the atmosphere. So it's a primarily terrestrial and aquatic-based cycle. These are elements, right? These are atomic elements: carbon, nitrogen, phosphorus. Those are elements of major importance to living organisms. They're critical to the growth of the vast majority of living organisms. There are other elements that are either less critical or critical in lesser amounts, or that get taken up by organisms but really aren't essential for their biology at all. Elements that exist in nature, perhaps in trace amounts, that may not be important for the functioning of organisms, the biological functioning and physiology of organisms, but they get taken up by organisms in the things they eat or the things they drink, and they get lodged in their tissues. As a result of that, we can use these elements as natural tracers of ecological dynamics. And the relevance of it here is that we can study these elements as they move through these systems today, but we often have access to these elements in fossils that record the ecological dynamics of ancient organisms. So by understanding the dynamics of these elements today,